So given the seemingly insurmountable odds in cracking the autism enigma, what drives scientists and doctors around the world to work on such a complex topic? Well, first, most of us, I think certainly all of us, believe that the truth of the origins of autism will be found in scientific investigation. So the truth will be found in science. Second, there is good evidence that early detection of autism, or ASD, and related conditions can benefit outcomes. Third, there is good evidence that a better understanding of the specific causes of autism will benefit the medical management of that individual. And fourth, given the heterogeneity, the extreme heterogeneity involved, individualized approaches may be the way forward in treating autism. And fifth, from the basic research perspective, to understand human development, namely cognition, speech, behavior, the attributes associated with autism, we need to pursue scientific investigation. On the right side of this slide appears the cover story from Canada's Globe and Mail newspaper. The article describes the discovery of the role of copy number variations, or CNVs, in autism, which our laboratory participated in. And in particular, in this paper, the identification of the neurexin-1 gene to be involved in autism in this family that is shown on the right. This type of excitement in the public often occurs when there's a scientific breakthrough that often offers hope for families. It's important to point out that ASD remains a behaviorally defined spectrum disorder, so the diagnosis is based on behavioral classifications. However, in the past decade or so, new technologies have been developed that allow the study of complex problems from the inside out, or the bottom up. Most notably for autism, the technologies that have had the most impact, arguably, have been in the areas of imaging and genomics and animal and cell models. Returning to the tree metaphor, these technologies have allowed us to see the trees within the forest in exquisite detail. In this vantage point of two trees, looking from the ground up, we can see how the tree branches interact with each other and their environment. I would like you to keep this image in the back of your mind, because in the next few slides I will come to talk about the issue of neurons and brain cells, dendrites and synapses, the so-called branches and twigs that I mentioned earlier. And I think you will immediately recognize that the images of the trees in the neuronal brain cells will be very, very similar. So as mentioned, autism remains a behaviorally defined spectrum with no effective drug-based treatment for the core symptoms of autism. Although great heterogeneity exists, the data from structural and functional magnetic resonance imaging, or MRI, techniques have converged to suggest underlying neuroanatomical atypicalities that produce an inefficient brain system, or perhaps in some cases an over-efficient brain system, when considering the complex integrative processing going on in the brain. This slide was put together by my colleague Jason Lurch, summarizing composite data from hundreds of different brain imaging studies of individuals with autism and controls, using different technologies and different testing strategies to define subregions of the brain involved in autism or aspects of it. And I would point out that actually much of the data was probably generated by people who are in this audience and participating in the workshop today. So I also just wanted to show this because this is a really, really cool image. So as we go forward, we'll see areas of brain involved. In gray coming up are the cortex and the cerebellum, which have been generally described to have a role in autism. In red, the inferofrontal gyrus, in red in ASD. Superior temporal gyrus in purple here, also involved in ASD. The cerebellum coming in green. And through testing in autistic individuals and controls to be involved in repetitive behaviors. In red, the caudate nucleus, also involved in repetitive behaviors. And coming in green, the thalamus, which is an important relay station in the brain associated with many different roles, also involved in autism. And finally, the amygdala in blue, which seems to have a role in the emotional processing attributed to a contribution to the autism spectrum disorder. So these are still relatively crude studies. The imaging techniques are getting better and better that even allow us to look at single cells and single molecules. And as I'll talk about a little bit later, I think an important advance will be to marry these technologies with the genetics and other advances. Albeit I'm biased because I'm a geneticist, in what I would refer to as the genetic and genomic enlightenment in autism, I would argue that the greatest advances in cracking the autism enigma have come through the study of families, chromosomes, and DNA. The first major advance came in the 1970s by Professor Rutter and his colleagues, who showed a strong propensity for monozygotic twins, who by definition have identical genomes, to be more often than not both present on the autism spectrum. This so-called concordance of autism between monozygotic twins drops off significantly when looking at dizygotic twins who share 50% of their DNA or genes. While different studies have found different levels of concordance between identical twins, the message is always the same, and that is that genes are involved in autism. As I would discuss, some of these genes have now been identified, and some of the mutations, a mutation is a genetic change that leads to a clinical outcome, are sometimes inherited, while in other cases they are new to that individual, and the technical term is a de novo mutation. What I find perhaps most interesting from these studies is that in some instances, even though the identical twin may be autistic, sometimes they vary and can be on opposite ends of the autism spectrum, one mildly affected and one severely affected. This observation tells us that sometimes non-genetic factors and perhaps even randomness can be involved in the process of developing autism. A second advance of genetics is that studies of families with ASD siblings have shown a higher coincidence of autism than would be expected by chance. The latest work, led by Dr. Lonnie Zwagenbaum in Edmonton here in Canada and colleagues, indicate that in roughly 18% risk of autism in second siblings of families with firstborn ASD children. And this is much higher than the predicted 1% general population level, again providing evidence that genes or genetics are involved. Their work and that of others has also shown that early identification of autism through the study of families can improve the outcomes, arguing that genetic diagnosis may have a very important role to play in early interventions. The third major advance of genetics, using the technologies I will show you on the next slide, is that specific deletions or duplications of segments of DNA encompassing genes, the so-called copy number variants or CNVs I mentioned earlier, and that are shown here in the second image in the panel in the middle here on this slide, or specific sequence-level mutations of genes depicted on the bottom image, have now implicated dozens and perhaps even hundreds of genes to be involved in susceptibility to developing autism. So we now have identified some of these genes that were hypothesized to exist. So presented here are the three major genomic technologies used to study DNA, not only in autism, but in fact in every genetic disorder. The first is a very old technology, some 50 years old, called cytogenetics or karyotyping, where you look at stained chromosomes and look at the gross morphology. And here you can count the pairs of chromosomes, chromosome 1 to 22, the so-called autosomes, and then the sex chromosomes, the chromosome X and Y. So this is the karyotype of a male. This is the technique that's used to, for example, test for trisomy 21 that's associated with Down syndrome. More recently, in 2007, what we call our microarrays or gene chips were introduced that have over 1 million spots of DNA on a glass cover slip the size of a quarter. And using this technology, which is a form of nanotechnology, you can take DNA from an individual and test it in a few hours to look for the presence and absence of specific genetic variants across the human genome. This approach really revolutionized the way we do genetic studies. It allows us to look at the whole genome in one experiment relatively cost efficiently. And the new technology is going to really change everything, and it is changing everything, is what we call whole genome sequencing using next generation sequencing technologies. So using this approach, we can now capture all 6 billion chemical bases of information in a single experiment. We don't have to test one gene of the 23,000 or 30,000 at a time. We can capture the sequence in one experiment, and in my laboratory now, we can do this for a cost of about $2,500 or so. The Human Genome Project cost $3 billion to complete. The first human genome sequence, which was of Craig Venter in 2007, cost about $70 million. And now we're doing it for $2,500 a genome. We expect it'll be $1,000 a genome. And when it comes into the cost range of karyotyping microarrays, it will become a standard clinical diagnostic test, and it's quite likely that each of us will have our genome sequenced as a step in personalized medicine. So these are the technologies we're dealing with. And here I have summarized research results generated over the past 30 years that have revealed a proportion, roughly 20% of the genetic architecture that is involved in autism. So if you were to take 100 individuals with an autism diagnosis off the street consecutively and run these different genetic technologies and also do kind of detailed clinical examination, you could group roughly 20% of those individuals to have a known genetic form of autism. And these include what we call ASD-related syndromes, occurring in about 10% of cases, including Fragile X syndrome, where we see autism in roughly 40 to 50% of cases, tuberous sclerosis, phenylketonuria, Rett syndrome, and I'll talk about Fragile X syndrome and Rett syndrome a little bit later, so please keep these in your mind. And there's over 100 different syndromes where actually autism is a component, in some cases, of the clinical presentation. There's also rare chromosome abnormalities detected by the karyotyping method I mentioned earlier. More recently, rare copy number variants I'll come back to in roughly 5% of cases. Chromosome 16p11.2 I'll mention a little bit later in others. And now using the DNA sequencing technologies, we can identify specific gene mutations at the sequence level, and we've now identified perhaps 100 or so of these genes. I won't have a lot of time to talk about it, but certainly we have now identified perhaps 20% or so of the genetic factors involved in autism. There's many different other explanations, including combinations of genes acting together, the role of other genes interacting with the environment. I'll talk about at the very end of my presentation, and also a type of a genetic modification called epigenetics, which you read a lot about in the newspaper, which is the process of modifying the DNA sequence based on environmental cues and factors. But the message here is that we do know actually some of the causes of autism by the study of these other syndromes and some of the genes that have now been identified. So I think it's important to show this slide to re-emphasize now that some of the CNVs, copy number variants, and genes that have been identified are considered to be the cause of autism in the individuals that carry them. These 6 DNA segments represent examples of the most common DNA regions found to be involved in autism in our Canadian population. Diagnostic tests for these CNVs and genes are now used in conjunction with standard clinical assessment. One of the complexities of the genetic findings is the fact that some of the same genes or regions of effect are being found associated with other neurodevelopmental conditions such as schizophrenia, bipolar disorder, ADHD, and intellectual disability. This observation is perhaps not entirely surprising because individuals and families with autism can exhibit these other types of disorders. And perhaps we'll talk more about this in the discussion. I will discuss the 4 genes or CNVs highlighted in red later with the SHANK1 gene mentioned on my next slide. So on this slide is an example of what we call a pedigree, a 4-generation family, with females shown as circles and males as squares. Individuals colored in black have autism, and all of them carry a familial or inherited deletion of 1 copy of their SHANK1 gene, which is typically present in 2 copies, one from mom, one from dad. And this is on an autosome, chromosome 19. So it's not the sex chromosome, it's not the X chromosome, and I'll talk about that a bit later. The index case, which is the first presenting case in the clinic, is shown with the arrow. And as can be seen, he inherits the deletion of SHANK1 from his mother. For those astute geneticists in the crowd, you'll probably quickly recognize that only the males who carry the SHANK1 deletion have autism, while the females do not. We don't know the reason for this yet, but it may be that females have other factors, such as gender-specific hormones, that protect them from the autism. The deceased grandfather, who also carries the SHANK1 deletion, is shown with a stippled box, since he had what is referred to as a broader autism phenotype, and this is a subclinical form of autism. So in this case, we could provide the explanation to this family why the autism came about in their situation. And I should say that we haven't seen this genetic change in well over 10,000 controls. So we use this type of population and family-based approach to convince ourselves that the genetic changes we're finding are in fact indeed involved in autism.